So I'm going to talk today about malaria and climate change, and I thought about it's complicated or complex. I don't know which, but it's both. And I'm coming at it from, I'm trained as an immunologist. So climate change is sort of, again, I think one of the things you kind of hear is that we cobble together our understanding and our studies of these different areas, not through traditional funding mechanisms, but just sort of gathering the data as we get it. I'll only give you a little bit of data, and I'm going to try to think about how we think about our data, how we do surveillance, and how that my scale of resolution is very different from a climatologist. I like this quote. Malaria is 1,000 different diseases and epidemiological puzzles. Like chess, it's played with a few pieces, but it is capable of an infinite variety of situations. And I think this is an important concept for us to kind of visualize about malaria, because it does have, it's very different than, say, from dengue or Zika or chikungunya, because, as I'll tell you, it takes a long time to develop immunity to disease, so you get repeatedly infected, and that's a very different piece of this puzzle. So if we look at the worldwide population at risk in 2013, this is the Malaria Mapper project by WHO. You can see areas where in sub-Saharan Africa, 80 to 100% of the population is at risk for malaria, ranging down to up in the drier areas in Africa, 0 to 20%, South America, China, and Asia. So we have this belt there that extends above the equatorial boundaries there, but areas where the mosquito that transmit malaria is present. I wanted to just give you 2 slides on background of the malaria itself. So there's 5 species of malaria that causes malaria, 5 species of the Plasmodium parasite that causes malaria in humans: Plasmodium falciparum, Vivax, Noelzii, Ovalii, and Malariae. The most common and the one that causes the most severe disease is Plasmodium falciparum. And this just gives you a sense of the numbers. This is in 2010, the number of cases, 216 million cases of malaria. So when we're thinking about scale of disease, that's quite a large scale there. And deaths due to malaria has actually dropped dramatically in the last decade due to a number of interventions, and about 655,000 deaths per year. And the most deaths are occurring in sub-Saharan Africa as well. So malaria is transmitted by the Anopheles mosquito. There's a number of variety of species of Anopheles, but and the different prevalence around the world. The mosquito bites you. The parasite goes to the liver, and it's in this liver stage. It'll stay for a period of 8 to 10 days. And we get confused between the 2 different types of malaria. I'll be talking primarily about falciparum, but the other parasite, Plasmodium vivax, is the one that causes relapsing malaria. And so it'll be quiescent in the liver, and then 30 days, a year later, 5 years, 10 years later, you can get a case of malaria relapsing occurring. But in sub-Saharan Africa, it's almost all falciparum malaria that occurs, and that means the parasite just gets into the liver, incubates there for about 10 days, and then emerges out of the liver, infects the mature red blood cells, and starts the cyclic infection of the red blood cells. At some point, we get this sexual stage development. We're not sure what triggers that sexual stage development. And the mosquito, female mosquito, has to get both the male and the female gametocytes to pick them up and have part of the life cycle in the mosquito. So when we think about malaria-related illnesses, there's a number that I like to think about, and I tell my immunology friends this. So when you're doing a blood smear to see if somebody's sick with malaria, you're looking at 5,000 parasites per microliter of blood, right? That's a lot of parasites circulating in your blood. It's a systemic infection. As we know, our blood goes everywhere in our body. Where our blood goes, so goes the parasite at that very high burden of disease. And this is just a picture of a child. You can see anemia. Again, it infects those red blood cells, so the red blood cells get destroyed and cleared. You can get severe malaria, including cerebral malaria and profound anemia. You can get acute respiratory distress. And then death occurs as well. And again, that death is usually due to the P. falciparum malaria. So the thing that I've been sort of thinking about with thinking about climate change and malaria is how do we, as an epidemiologist, and I would call myself an immunoepidemiologist, think of scale and study malaria. So if we go back to this slide of the worldwide population and focus here on Kenya, which is where our research program is in East Africa, we work at the Center for Global Health Research at the Kenya Medical Research Institute. We have actually very nice laboratories there. That's my now postdoc, Sydney, in the laboratory. This is at the CDC building there. It's a lovely spot and close to the populations that are sick with malaria and where we can do biomedical studies. So early on, and I came to malaria actually by studying a children's cancer called Burkitt's lymphoma and linked to EBV. So it's a long way around to wherever we get to our studies. But we mapped out within Kenya, using data from Bob Snow, different levels of malaria endemicity. So up in there's a low-risk region here. There's arid seasonal up in the areas near Sudan and Somalia. Along the coast, you get sort of endemic coastal malaria, lake area along Lake Victoria, lake endemic, and then highland malaria. So we have very different, so even within the country, where you can see it mapped all brown in one study, when you actually start mapping it and looking at it more closely, you can see there's variations in the levels of malaria transmission. And this just wants to show you, we were looking at two sites, one in Kisumu and one which is along the coast in this lake endemic region, and one in Nandi. And I just want to show you, this is age group and children. It was a cross-sectional study of kids between 1 to 4, 5 to 9, 10 to 14 years of age. And this is just to take a look at the subjects with Plasmodium falciparum positive blood smear. So this is just detecting malaria by looking at the blood smear. It's 77% of these kids were PF positive. Now, the other thing is these were healthy kids. These are not sick kids. These are ones, that's what we define endemicity as the percent of parasites in children where you don't have evidence of disease. And you can see their mean body temperature was just fine. If you just go up to the highlands, which is about a 2-hour drive up to Nandi, you see a very different scenario with average about 16% of these children are malaria positive. So on that scale, in Kenya, just going a few hours away, you see some very big differences. If we look within Kisumu County, which is where we've been working, and this is Lake Victoria there, we were mapping out where cases of Burkitt lymphoma were, and we identified areas where Burkitt lymphoma was higher than expected and areas that was less than expected. And so I had a student, Odata Sumba, who then wanted to look at what the difference was risk between these sites, and these are 6 miles apart. So now we've moved from a 2-hour drive down to 6 miles within the same province, same ethnicity, same, pretty much same everything, except within this small region, the area that had less than expected Burkitt's lymphoma, when we did a cross-sectional study, had much less malaria parasites. So very small, that's 22% there versus 62%. So when we think about this geography of malaria, it's very different in even microgeographic regions. And so the percent of children, what we consider in some ways are not yet immune, and I'll explain what I mean by that have parasites in their blood is an indicator of how much malaria exists within a region. So when we look at this, where we've heard a lot about the prevalence of the mosquitoes, but we also have to look at the susceptibility of the host and how prevalent that parasite is within the host. Now, one of the unusual things about malaria as an infection is that you can get repeatedly infection, and this is just a cross-sectional study. This is one kid in our study where we're looking at blood smears, so the parasitemia, parasites per microliter of blood. Not very high, but this kid, probably about 4 months of age, had a malarial infection, and then again at 11 months. This is a child born in Kisumu, where there's a lot of malaria, and so should have been protected by maternal antibodies and wasn't. But what you see when we looked and measured, and this is just measuring antibody responses to 2 antigens, things that malaria makes. So these are things you should be fighting off malaria and making these antibodies. You see you get a nice peak there, and then you get a nice peak there. But from an immunologist point of view, what should be happening is the first time you get infected with something, you should make a little bit of antibody. And the second time you should make a lot more, and it should be a lot faster, and it should stay on. And these antibodies don't stay on. So you have, it sort of fails 2 of the common themes of immunology about how our immune response. So when we're thinking about vaccination, the key theme of vaccination, you get a measles vaccine that protects you against the measles because you get that first non-lethal form of measles. And so when you see it the second time, your body responds very rapidly and makes a good response. That's not true with malaria. The other thing we see with malaria, this is just a panel of different looking at antibodies to different malaria antigens, but I want to just point out this is looking at in kids less than 5, 6 to 15, and greater than 18 years of age. You can see that the amount of antibody takes a long time, so this is just levels of antibody, so the 5-year-old, the 6 to 15, and the 18-year-old. It takes a long time for them to make immunity, so you eventually develop immunity to the malaria pathogen, and what you do is you actually develop immunity to disease, not to infection, and that's why we can do these cross-sectional studies and see how much parasite is there, and those kids are healthy, although now there's a lot of data indicating they're not as healthy as we thought they were. But these kids, once you make it past the age of 5, you're able not to get the disease, but you still get the infection. So the point here is that we have geographic variability of malaria prevalence even within a small region. The percent of children that the not yet immunes is an indicator of how much malaria exists, and immunity is slow to develop and only protects against disease and not to reinfection. And in these cases, we think about it's a chronic infection. So when you look at that map of Africa and sort of think about it, when you think about that host, the people that are susceptible, it's more of a chronicity of an infection rather than these acute infections we're talking about like with Zika or chikungunya. And I took this map from a paper by Martins and Thomas looking at different temporal and spatial scales that affect malaria epidemiology. And so they have 1 centimeter, 1 millimeter all the way out to the earth, and then hours through millennium, so this log of a temporal scale. So they think about the mosquito, the larval development, and the adult mosquito. Think about how weather might be shaping this factor. I'm not sure why they threw in the human migration, but they did. Moving into places where you might, or moving out of places where there's malaria. El Nino and global warming and how those all play in together. But what I would add is we have to think about the transmission of how does those mosquitoes get to the humans and what do we do in terms of our immune response to that. And so that transmission and immunity are two things that sort of have to be considered as we look at these different types of models to make these predictions, because without having the host piece, and there's one other piece that I don't even mention, which I will now, is the human genetics and our evolution to evade. Malaria has been an incredibly driving force of human evolution. Sickle cell is thought to be an evolutionary mechanism to evade the malaria pathogen, and a lot of these other hemoglobinopathies. So we have to take those factors into consideration as well as we try to begin to understand how climate change can affect malaria. And so I took this from a recent paper, Earth and Planetary Sciences, Impact of Climate Change on the Geographic Scope of Disease. So here's the map of the current distribution of where malaria occurs throughout the world. And then in red is showing possible extended distribution by 2050 in terms of suitable climate. And again, we also have to think about not only suitable climate, but bringing up the question of interventions as well. And so the models that are out there right now hold true only if no actions are taken. So that's another important point to think about as we look at these climate models on malaria. So intuitively, we know that if it's warmer, there's more mosquitoes, they can move to different regions. But we also know that with malaria, we actually have a lot of good things in our toolbox to prevent malaria right now. And this is a great figure. This is from the Malaria Atlas project. This paper was just published recently by the group from Oxford who's been doing the work. This is a map of malaria distribution in Africa in 2000, and this is 2015. The darker colors are the more intense transmission, and you can see the fairly dramatic changes in malaria transmission intensity over the last 15 years. And a lot of this is through intensive efforts for a number of organizations and groups and countries. Distribution of insecticide-treated bed nets. The mosquitoes that carry malaria are night biters. They bite you at night. So if you have bed nets, you can prevent the biting. They've been doing indoor residual spraying in the houses, very active spraying, so you get rid of the mosquitoes that are inside the home. And then before the 2000s, there was the emergence of resistance to chloroquine, which caused a resurgence. One of the reasons for resurgence of malaria, certainly in Africa. But the development of what's called the artemisinin drugs has changed that, although we're also in danger of developing drug resistance in that regard. And then lastly, I wanted to bring up these two papers. One's by a group by Guy Yanyan and Andrew Gatheko that work at Kemry and SUNY Buffalo, now UC Irvine. And then the group Simon Hay and Bob Snow at Oxford. And this was sort of a spat that played out in the literature. And in fact, I found a quote from Jonathan. I didn't have time to throw that in there. But I think this was 2005, they published a paper saying that the Kenyan highlands and the East African highlands, and so this is the one place, Mount Kilimanjaro, Mount Kenya, so we actually have a little bit of snow there. Andrew Gatheko, there's an anecdotal story where he started noticing that there's more of the Anopheles mosquitoes in the place, his homeland, where he was going back to. He'd never seen them. There was an outbreak, an epidemic of malaria. And so they went to do some surveillance and did some studies to show that there was an increase in malaria prevalence, both the presence of the mosquito and infection, and linked it to climate change differences that allowed the mosquito to move up the mountain. However, this group here, and Simon Hay and Bob Snow is the one that gave us our data in terms, and I'm not taking sides in this, I'm just pointing it out, but he gave us the data. They'd been mapping malaria in Kenya for a long time, had a lot of years of data, and they argued is that malaria epidemics in the highlands of East Africa garner significant research attention, due in part to their proposed sensitivity to climate change. In the recent article over here, they claimed that increases in climate variance rather than simple increases in climate mean values had an important role in the resurgence of malaria epidemics in East African highlands. If proven, this would be an interesting result, but, that's always the but, we believe that the methods used do not test the hypothesis suggested. And so this is controversy between is malaria increasing in African highlands and what impact that would have. I've read some articles saying that just 2% of the population live in the highlands anyhow, so putting the resources towards that doesn't make sense. But the other piece is coming back to the surveillance and thinking about with dengue, Zika, waterborne diseases, how do we know how much disease was there? How do we measure that? And then how do we get the impact? And I think as we move forward, one of the things is making sure our surveillance tools are in place and strong, because I think the challenge with this paper, is that I don't know if they had the best data in terms of collection of malaria prevalence, historical data to grab and analyze that. And also the time scale that they were looking at was a period where we also had resurgence of malaria because of the chloroquine resistance and other factors as well. So I think that I just wanted to highlight that as we try to look at these questions. And it's not, I wouldn't argue that climate change might not impact malaria, but that it is complicated as we try to understand all the different factors that play into emergence of a disease and spread of a disease in different populations. So one of the things, sort of my unanswered questions was, and this is what I've been thinking about, is to bring the scales of climatology and malaria epidemiology together to increase our predictive and analytic power. From the malaria epidemiologists, we really need to work on having robust surveillance so that we have good, accurate data in places that don't typically have it. I would say that when I see maps of Africa, and somebody I was grateful pointed out, just because there was gaps there doesn't mean there's no disease there, it's just because we don't have data. And I can tell you that getting accurate data in resource-poor countries is very difficult. And then to make sure that we think about how interventions affect our modeling of disease so that we include that in those interventions as we try to model disease risk. And then lastly, I'll just raise it because we've been talking about kidneys and kidney disease. Does malaria contribute to other chronic kidney diseases? And so I'll end with this article about, from Sri Lanka, dehydration and malaria augment the risk of developing chronic kidney disease in Sri Lanka. So one of the things when we think about malaria, and this was sort of the question Henry asked earlier about dengue or other ones and bringing up, do we have infectious diseases that exacerbate this acute kidney injury? And in the case of malaria, we know it does cause, when I talked about the systemic infection, you have acute respiratory distress, but you also have kidney problems with malaria as well. And so I just put this in there to think about dehydration, history of malaria, and drink and untreated well water are likely contributors. Now, a history of malaria is very difficult to have. And again, that's getting that accurate malaria data and making sure we understand what they're gathering there. But I think it's worth thinking as we talk about climate change and we think about chronic kidney disease, if malaria becomes resurgent or spreads to other areas, it might contribute not just to malaria-related illness, but also to other ones as well. So I will stop there and take questions.
I would think that during the colonial period in Africa, there would be more data than post-colonial, because there was such, the British and all the other Europeans data there.
Yeah, so it depends on which colonial group that was there. So Kenya had fairly good infrastructure, but Kenya also had infrastructure relative to places that they cared about. The highlands where the tea was, they cared about that. And the other piece is that it's before computer records, right? So if you think about that, it's all paper-based records and mostly blood smears. And then I'll just say, even in the highlands of Kenya, when I was there in 2001, I brought a microscope up because they had an outbreak, but they forgot that they had no power to the local clinic, so they couldn't use the microscope, but then they got the microscopist. So we don't really have good past data that has been accessed.
In climatology, when we don't, when we go back in time, we don't have the observations. We use paleo records, right? Is there an equivalent way of getting some proxy, to try? I mean, it's sort of a general question you've got to think about, but I mean, it seems to me that there may be something that could be used as a proxy to have some estimation of what may have gone before.
Well, that's a good question, and I'm going to have to think about that one, how you could take a look at that. You could think about sort of suitable environments. Maybe you could look at the record of plants in a particular area, but that'd be challenging, but we're thinking about. Because at scale, right, I'm working within a year, 5 years, and 400,000 years is a big gap.
Well, we wouldn't be going that far, but just, on the order of 1,000, 2,000 lakes, for example, we have may show something.
Jonathan?
I have a comment about the highland malaria, because I was in the middle of that, got in the middle of that tangle. 2 comments. One, it's for the climatologist, and it relates to some papers put out by Madeline Thompson and then Mercedes Pascal, showing that, especially Mercedes' work on how much temperature change does it take to affect Anopheles Anopheles Fenestis and Gambii, how small amount it takes to completely change the population dynamics. And the bottom line is that you don't need statistically significant temperature or weather changes to actually drive a very significant change in mosquito biology. So that's one message. The other one is that malaria is complicated, as are all these diseases, and there are some of these, and I think some diseases will be indeterminable regarding long-term climate attribution. Malaria may be one of these where you don't have multi-decadal quality databases and no in and out migration, drug resistance, pesticide. It may be impossible to determine, full attribution in climate change links, but that's why we rely on the short-term El Nino and other variability stories. But anyway.
Good point. Andy?
I think that's a really good point. Kind of what type of success will we have in kind of attributing climate change impacts on vector-borne diseases, and to what extent do we even need that? I think being able to project maybe changes in suitability for transmission, which generally for most vector-borne diseases, as you know, is that it would be more suitable. Is that enough to implement the, what are you trying to do, what action are you trying to influence by doing these projections? And I think, you're trying, perhaps you're trying to influence near-term interventions that can build capacity to address long-term and short-term vector-borne disease threats. And so I think it's an interesting question of, how much effort do you need to actually put into that endeavor, given that the probability of success is low, I think.
Yeah. This is just another aside on that same, on the Highlands story, right? I think Madeline's group showed that some of the weather stations had been moved, and when they did these adjustments in the climate time series, the story changed completely. So it's not just the malaria data, but the climatologists in the room know that it's difficult to work with these long time series as well. Someone can move a weather station here and not write it down, and it can look like climate change.
Just thinking about the comments from Jonathan and Andy that regarding what information can we know, what information do we need to know. A lot of our discussion has been about trying to anticipate a problem, but or a trend. I think what malaria brings to mind is the fact that not only do you want to anticipate or observe a trend, but you want to intervene. And I want to strongly endorse what Andy just said about that, because in my arena, which actually is occupational medicine, it's much more important for us to know whether asbestos is present or not than whether we can count lung cancers from asbestos. If we reduce the asbestos exposure, we can assume we reduce the lung cancer risk. And it seems to me we have to be thinking in those terms rather than trying to always complete the cycle as long as we have enough information about relationships.
I think this is this really fundamental point that you raise, and I have a lot of tension with it in my own research, in that to some extent we know what we need to do for malaria and diarrheal diseases, and we're not doing it, or not doing enough of it, because there's interventions that can reduce this, so we don't need to talk about climate change at all. But on the other hand, if it's going to, it is our role to talk about mitigation and really link these really devastating health impacts to emissions, or is our role to help with preparedness and adaptation? And I think, depending what our goals are there, I think the potential models and outputs would be different. So is it, how far upstream are we going on the intervention?
The other thing that's interesting is, these papers that come up that show that fevers and viral infections can be associated with acceleration of CKD. And, trying to understand the mechanism, whether it's like simply fever or is it muscle lysis of red cells and uric acid load or this or that, those kinds of things, is really an interesting pathogenesis question that if we could get to that, we might actually learn more. And it's interesting, we can talk later because I'm not going to talk about it tonight, but evolutionarily, there's a link with malaria and the pathways that I'm going to talk about tonight that's been written or postulated in the literature. So it's kind of interesting that, the innate immune response to malaria involves some of these, some aspects of what I'm going to be talking about tonight.